Hello everyone, and welcome to today's webinar, EBSD-based correlative microscopy. Before we get started, I'd like to cover a few housekeeping items. If you have a question, please submit it through the questions pane on your console. Any issues regarding connectivity and webinar viewing will be addressed immediately. We will attempt to answer any questions for the speaker at the end of the presentation. If we do not get to your question, we'll follow up via email after the webinar. Now I'd like to introduce the speaker for today's webinar. Stuart Wright was first introduced to EBSD as an undergraduate student at Brigham Young University, working with the first system in North America. He then moved on to Yale University to pursue a PhD under Professor Brent Adams. His thesis research focused on automating the EBSD technique. This effort led to the first fully automated EBSD scans in the fall of 1991. After graduating, Stuart joined Los Alamos National Lab and continued work in microtexture and texture analysis using both the OIM technique and conventional X-ray diffraction. Shortly after TSL was founded, Stuart left Los Alamos and joined the startup company commercializing the OIM technique, working primarily on software development. He has continued in this role through the purchase of TSL by EDAX and then Amatec. The original OIM system at Yale could index approximately one pattern in 3 seconds. Modern systems have come a long way since those early days, with speeds now exceeding 6,700 index patterns per second. Stuart is closely involved in the continued development of the technique, as well as in working with scientists all over the world in applying the technique to their materials research. Now over to Stuart.
Hi everyone, it's good to be with you this morning, or whatever time it is where you're at. Today we're going to talk about correlative microscopy, and this is very much going to be a how-to webinar today, which is a little different from the ones I've done in the past, but hopefully it's helpful to you. We'll go through a brief introduction and also how we did this in the past, and then how we are going to do it both in the current version we have, but also we've added some more tools in what will probably be a 9.1 release in the near future. And I'll show you some how-tos to do that with examples from various techniques. I'll have examples from micro-Raman, micro-hardness, ECCI, DIC, WDS, cathodoluminescence, optical line microscopy, and a couple other examples. So if you look up the word correlative microscopy and go to Google and find something, I found this nice statement that comes from actually the National Institutes of Health in the United States. It says, correlative microscopy is a method for the analysis of the very same cell or tissue area. Several different methods of light or electron microscopy are used. Now, I don't look a lot at cells or tissue areas, but we can use it to look at microstructure. And this is an example from a paper by Daniel Abu-Ras, and he shows an EBSD map there on the left, as well as the second one there, the EBSD pattern quality. But then he uses EBIT, cathodoluminescence, and in that paper he looks at some others as well. And you can, if you look at those closely, you can see those are the same area. And that's kind of what we want to do with correlated microscopy, is to get different views using different techniques of the same area to better understand material behavior and how our microstructure will influence properties. I recently finished a book called An Immense World, and it's the idea of looking at how different animals look at the same environment. We all know about bats and their echolocation skills, but reading the book, I learned about how some animals can sense electric fields, magnetic fields, mechanical response in terms of vibrations, infrared radiation, and they all create an image, essentially, of the same thing that you and I would see with our eyes. And when they do that, then they can better understand why animals behave the way they do and understand information they see that we don't see. And the same is true with correlated microscopy. We get different views using different techniques so that we can better understand our materials. So we're going to use as our base image, not our eyesight like we would in that book that I read, but using EBSD. So we want to correlate other imaging modalities with EBSD results, whether that's EDS, which we've done for a long time to get the chemical composition, or it might be micro-hardness, or it might be other techniques. So our very first foray into this was with EDS, that makes sense. And we wanted to correlate EBSD with EDS. And we did this in an iron nickel meteorite sample, and you see there on the left is the SEM image, in the middle is the nickel counts, and on the right is the IQ map. And so we were able to correlate the chemistry with the EBSD, and we did that at an M&M and showed those results a long time ago, 1997. And we've come a long way since then. But there are, even at that point, we realized there are some challenges. There's different imaging instruments. So EBSD is done in the SEM. We might want to correlate data from a TEM or light optical microscopy, or even something like a synchrotron or something like that, or mechanical systems. So AFM, for example. So that poses different challenges because we have different geometries. With EBSD, as you know, we're highly tilted, and most other techniques are done flat. And so sometimes that creates some challenges to correlate because of the different geometries. Other differences between different imaging modalities include spatial resolution. Obviously, if we're in the TM, we're at a higher spatial resolution than we would be in the SEM for EBSD. The depth of information. EBSD comes from just below the surface. It's quite surface sensitive, but we do get some information from below the surface a little bit. Whereas TM, you may have information coming from the top or the bottom, depending upon the technique. There are sampling differences in terms of when it was done. You know, can you do it in situ or can you do it ex situ? Some are destructive, so you have to think about which technique you need to do first. And then obviously we talk about correlation challenges. So you have differences in size, you have various distortions that can happen. When we work at high tilt, we tend to get some distortions. You may have the data rotated. Obviously, there's just simply when you put stuff in an instrument, when you put a sample in an instrument, you never get it exactly the same, so you may have to deal with that. And so hopefully it's just shifts, but sometimes it's also distortions that have to be dealt with. Ooh. Sorry about that. So our old methodology was to, at least this is what I would do, is I would take the text file, an ANG file that can be exported from OIM, and I would steal the IQ column of data and replace it with some other column of data. And that's how I actually did that original chemistry work where I showed the nickel counts. I just replaced the IQ with nickel counts. And that kind of worked because you can manipulate things like that in a spreadsheet editor like Excel or something like that. But as we go to million point data sets, that becomes really cumbersome and it's just not a great way to work. So the next step we did was to, you can use a map, you could export the map values and then take that text file and do kind of the same thing. You can replace whatever field you created the map with a new one. You could bring it in when you import the data. You could bring it into a custom value that the OIM software knows about. So you could do some correlated microscopy that way. But, and then you can continue to import and export and use this custom value to create your maps. And just like, here's one example where we exported some orientations and patterns into HR-EBSD codes into the cross-correlative microscopy that is used to look at strain. And then we would import it back into OIM so that we could image it in the same way. So you see there on the left, there's a CAM map out of OIM. We imported the data back in, where we measured GNDs using the cross-correlation results. This is work that Tim Ruggles did, and you can see the paper there. And then he also had some ECCI electron channeling contrast images in the paper as well. So you can see the correlation between those images, and that does provide some interesting data. These are all very, very related techniques, and so they correlate quite well. So our goal is to do this in different ways. And so we still have some text-based import techniques, and so I'm going to start with those, since that's the way we've done in the past. So this is an example here where we use EBSD and cathodoluminescence. And we also had some different IQ measurements that we've used in the past, image quality or pattern quality measurements. We use one based on the Huff, but you can imagine some based on the patterns themselves. So here's some just examples of calculating those. And so the first step is to load the EBSD data into OIM using the standard way. And then there's an icon at the top. I should go back here. If you look at the software, there's a little icon that looks kind of like a light microscope. I hope that's what it looks like. This was my idea when I drew it. And there's a pull down, and you can import from a text file. So you can see here, there's a little one I call iqstudy.txt, and you can load that in. And you are required to have columns for X and Y data. You can see the first 10 lines in the file there at the top, and you can see then the X and Y, and then the next step is the deviation version of IQ, the entropy version, and the mean version. And so I can add those in, and you can see then those are added in to the software, and then I can create a map. And what you do is you will see a correlated microscopy value there in the properties. I'm doing the grayscale there. You can see the value there. And so the next step then is to just simply make a map. And so you can see here, the top left is the standard IQ map. The other three are my correlative microscopy maps. So they aren't really labeled in any way there. I've just called them CMV maps. I'm not sure which one I did. I neglected to change the names. But you can see the correlation with the original IQ map, and that they provide slightly different information. They're just based on the patterns themselves. But the point is that you can bring in this data this way into the IQ maps, or into these CMV, correlated microscopy value is the idea behind CMV, and you can get the correlation. So if you can make a text map, and it doesn't have to be a one-to-one relationship. If I go back, you can see in this little chart here, I have an XY, and it will map whatever XY values you have onto your data set and find the closest point in the EBSD data and map this new IQ data onto those points. So it doesn't have to be a one-to-one relationship to what you actually collected, which is helpful. You can make a one-to-one, so if you want to do your own stuff outside to do that mapping, you can do that and then bring it into OIM for imaging. But we want to look at some other techniques. So I'm going to show you once again, this is a CL example, data collected by my colleague Matt Nowell. Oh boy. This is a cadmium telluride solar cell material, and data was collected on the Gatan Monarch Pro, and you can see the details there. We found a peak at 807 nanometers. You can see about 26,000 counts, and the EBSD data was collected with the velocity super. And so in this case, the images are already aligned, so it's a pretty direct import, which is quite helpful. Makes life a little easier. So once again, we load our EBSD data we collected. So you see that there. And then we are going to import from images. So we have on the left is an image coming from EBSD, and on what we're going to do then is hit an add button. And we have two images. We have this the image from the cathodoluminescence for the 807 nanometer peak, and we also have, this is quite helpful to us, the SED, the selected or the secondary electron image from collected when we were collecting the CL data. And so that pops up a little image that looks like this. We say great, and we bring it into our correlating software. And you can see here that for one thing you'll notice is that the images are quite a bit different. The one on the left, once again, is the IQ. The one on the right is the CL. And we need to correlate those. Now we know in this case it was done at the same geometry, so it's just a direct correlation, so that's pretty handy. I set my transform method to direct. If I hit preview, then you can see what that's going to look like. It's direct, so it's pretty easy. And we commit it then to our data set, to our EBSD data set, using this little commit CMVs to dataset, add button. And then I get my CL data into the software. So when I go to make a map, I can see a correlated microscopy value there in the middle image, and then on the right, I get to choose which one. In this case, there's only one. It's the CL at 807 nanometers. And I press OK, and there in the color, now I have the CL data, and I have behind that, I have the image quality values from the EBSD. So now I can correlate the wavelength emission at that peak to my EBSD data and look for any correlations that might be there. In this case, there weren't any real strong correlations, so I don't have really anything to show you there, but it's a nice example of a direct import. Here's an example for WDS, and this one takes a little bit more work. We did the WDS flat, and we collected, in this case, we have maps for aluminum, lead, and silicon. And you can see the EBSD then data there to the left. And it correlates pretty well, especially if you look at the silicon map. You can look at that versus the IQ map and see how those two images correlate. So let's bring that data in. So once again, I import it from using the CMV tool, and I select 3 images in this case. And you'll notice that they have different color scales. So the aluminum is a cyan or bluish one. The lead is the tan, and then the silicon is a purple magenta type image. But we can deal with that. So the one thing the import software does is it tries to guess at the gradient or the color scale. And so 100% is, in this case, the lightest color that can be found in that image. And then we assume it goes down to black in this case. We look for the lowest and create a gradient. And you can then give those numeric values. So in this case, we just say it goes from 0 to 100. I don't have anything information about the WDS counts associated with those, so I'm just going to use this 0 to 100. So I know this is the aluminum one. I bring it in, and I do that for all 3 images. Just make sure the color scale works, and it pulls into the correlating tool. And you can see there on the right, it just brings in a grayscale image into that tool. But you can switch it back to color if you'd like. So in this case, it's not a one-to-one correlation. It's not a direct correlation between the 2 images. So we're going to have to correlate the 2. And so we're going to use, in this case, what we call bilinear correlation. And you're going to have to click on pairs of points between the 2 images. For bilinear, you need at least 6 pairs of points. So here's a bunch that I've chosen. In this case, I ended up picking 9. And hopefully you can see then the correlation between the 2. And then when I push the preview button, you see what's going to come into OIM. And I'm happy with that result. And so I just go ahead and press the add button. So sorry, I've got some things hard to see on my text here. Okay, so when we work with color images, the pairs of dots can be tough to see, and that's why we've added the ability to do grays. And you can switch back and forth between color, gray, or an automatic contrasted grayscale map. And that makes it easier then to see the points and make sure they match up. And once you're happy, then you can bring it into OIM. And so here is a WDS map for the silicon on top of the image quality, and that shows you the match between the 2. That's a good correlation. The transform we come up with to bring the 2 maps into correlation is quite good. You can see the silicon map or all 3 maps actually, are the EDS map or WDS maps are slightly smaller, and so you have some areas you don't cover. And just to show on the right there, I've mixed all 3 together to make this nice little blended WDS map. And so that allows us to correlate these, and we can do all kinds of things to better understand the correlations between the EBSD data and the WDS data. So that's another example. Now we want to look at micro-hardness. This data came to me thanks to Chava Nakash Naik in University of Hyderabad, where it's a fairly new paper correlating EBSD with micro-hardness results, and it's in a dual phase still. And the data was collected using an EDAX. The EBSD data was collected in an EDAX Velocity Pro, and the micro-hardness data was done using nano indentation mapping. And so the figure at the top right then is from the nano indentation mapping. You can see the map that they end up with and the hardness function XY, and then you can see the EBSD data. And it's kind of hard to see how these two images correlate, but let's see if we can figure it out. So once again, I bring in the image, and now this is a little trickier because we don't have a kind of monotonic color scale. We go from, in this case, to almost a black to red to yellow to cyan and blue, so it's not clear how that should work. But if you remember, if you look at the right, they gave me the figure had the actual color scale on the right there, so we can load that color scale in. I just cropped it off using a simple graphics editing tool, and then I can load it into OIM, and then I can change the color scale, and you can see the values now are hardness GPA from 2 to 8. And so if I do that, I can start to look for correlations. It's a little difficult given these two different images because the grains aren't totally obvious, but when you look close, you can see. So I've clicked on, looks like 8 different pairs of points between the two, and you can go ahead and when I preview that, it worked out, and I can bring it into OIM as you see there on the right. So you can see the areas in blue with, in this case, they have the lower hardness versus the ones in others. Oh, why does it keep happening? Sorry, I got a new mouse and it has some buttons I'm not aware of. So that's a really nice example of using images to do the correlation, and just to show that we can correlate images from all different types. So let me now show an example where we have results from an electron microprobe and CL. And this data comes from Guillaume in France at the French Geological Survey. It's on a mineral called cassiterite, which I don't know much about, but the images are beautiful that Guillaume found. And so the EBSD results are from an EDAX DigiView camera, and we have CL results as well as electron microprobe results. And Guillaume did a really nice job correlating these in his paper, but I'm just going to show you, using his own techniques, I'm going to show you how we would do this today in this new version of OIM. So let's start with the CL. So you can see here the EBSD map. You can see the CL map. The nice thing is we have these kind of fracture lines, and so you can see how they correlate between the two, especially if you look at the secondary map, which accompanies the CL data. So once again, I can import these from images. You can see that I'm going to import the CL and the SE maps into here. And I can switch back and forth as I do this correlation. It doesn't matter. So if there's some feature that's easier to see in one map versus the other, I can switch back and use that one. And that's what I did with this, because there are a couple of interesting things between the CL data and the SED data, or between those two images. And it wasn't always clear to me which ones I should use, but I was able to do it. So if I correlate those two, then you can see I did, like I said, like I did on the others, I did 9 points in this case. You can see the pairs there on the left that I used. And I was able to correlate those, so you can see the top row is the EBSD with the secondary image, and you can see how well they line up. And then I did it also with the CL. You can see how they line up. And you can even then start to correlate different techniques. So you can see the IPF there with these other techniques and see how they correlate. And so that worked really well with the CL data. The EPMA data was a lot harder, but let me show you the results from that. Once again, in this case, I've selected a whole set of images, looks like 8 in total. And I had to go through they all use the same color scale, but obviously it's not, once again, it's not a simple one from in black to white or something like that. So once again, I was able to import the color scale. And then you can see for each one of these different images, the microprobe analysis images, you can see that I had to enter different values for the minimum and maximum values for the color scale. That's all doable. It's a little tedious, but I was able to do it. And I changed the labels so that they were a little clearer once they were imported into OIM. So did all that work, and you can see then the points I clicked. It was a lot more difficult, but because there were so many different images, I was able to do it. And you can see, even looking at this, you can see there's a rotation between the two. And realizing there was a rotation quickly helped me find the pairs. So there's the CL data, EPMA data, and the EBSD data all correlated together. Now, one of the things with this was a little bit more challenging than the others, and we also have a tool so that you can go back and say, okay, you know, the pairs I used weren't great. Let me try some more pairs maybe, or let me edit the pairs I have, the correlated point pairs. And so there's this function you can see on the left. It's located off the right mouse click off the edit CMV button, off the right mouse click off the data set. And you can edit individual ones if you use multiple image sets, make changes. This one, for example, let's say I got the scale wrong. I could change those scale values and get them correct. And I don't have to redo everything. I don't have an independent image of the micro-Raman results that were also in the paper, but here's a nice one in the paper that I referenced. Really beautiful results they obtained. So here's another set of results. This is digital image correlation, and I hope each step on these, I show you a different way that the tools can do things. So some of the data, the EBSD data comes from David Fullwood at Brigham Young University, and the DIC data comes from Zishan from Sam Daly's group at UC Santa Barbara. And there's the paper there if you want to get some more information on this. Some really beautiful images in that paper. Now, and this data was in a binary format. And so I want to show you how we were able to import that. You'll notice on the right, the image there, and you can see how the two images correlate, how that DIC image correlates with the EBSD data. So this time we're going to import from a binary file. That's the option we're going to use there. And it's, in this case, it was a MATLAB file that we converted to a raw file, just basically a binary bit dump of the data. And here's the figure on the right, shows how the data is written to the file. So you can see the first two rows of data, or first two entries in the binary file are basically are the size. And then after that, it's each set of data points. So we have various data points. So there's two ways that data can be put, that we can handle data formatted in a binary file. The first one is planar, which is the way this is done. Each type of data, each basically image is written out one after another. There's also packed data where you kind of have a structure and you write it out all simultaneously, kind of like a set of columns, if you will. And so you might need to know the offset, the gap between the slices, that kind of information. But those of you who are familiar with ImageJ to load in images, it's kind of the same idea as that. It's kind of how format we borrowed. So the first thing I need to enter is the width of the data and the height. You'll see there. This is quite a bit more than our EBSD data, just so you know. The offset then would be two integers, and so I need to account for that, which is 8 bytes there. And then there's no gap between the slices, so that's pretty straightforward. Now, the first thing I need to do, I need to figure out what data I have here. So in this case, I've got, we're gonna skip some of this, but because it comes in an order, I have to know that. So I'm gonna skip this first set of data. I'm gonna keep the next set of data. You can see the images from the data there. And then here's the results. Now you'll notice that the way the data came in, it's rotated 90 degrees from my EBSD data. So I use a little tool there in my import tool to rotate it 90 degrees, and I apply that rotation to all the images. And now you can see I have my EBSD data in the left for my correlative technique, my DIC data on the right, and I just click on some points, and this time I did 11. It was pretty easy. I just used basically triple junctions and was able to correlate those. And you can see it's pretty obvious that I'm just going to get a little, the DIC, even though there's so much of it, it's going to get mapped onto my EBSD grid. So I am going to lose some detail. So this shows the EBSD boundaries on top of the DIC results. But it's a pretty nice correlation. And this is, I was curious, so this is showing the Taylor factor with the basically the slip lines, if you will, from the DIC data. And you can do further correlations using that data, statistical analysis, but I won't go into detail how to do that today. The next type of images that we can bring in are polarized light. And this is a little different approach. So once again, I'm going to use the little tool. This data comes from Salzgitter-Mannesmann portion in Germany from Marco Ritter. And in this case, we're not going to actually associate a floating point value at each data point like we've done, but we're going to associate a color. So we've had to modify OIM to allow colors at each point instead of a floating point value. But that's been done. And it's done quite similarly. We bring in the EBSD data, and we bring in images. In this case, I have 3 different images I'm going to bring in. There's no color scale involved because it's just color. It's not associated with any kind of value. If you know some kind of value, then you would bring this in a different way. But in this case, we're just associating with the color. And once again, I do click on the points. And you can see each time I've used bilinear here, we also have a quadratic fitting if there's a lot more distortion. To be honest, the images I've done so far I've been able to do with bilinear just fine. And the quadratic, I haven't really used it very much. So I have my 3 images. In this case, it was a lot easier to work with them in grayscale than color for whatever reason, but I was able to do that. Oops. What I found in this case is that I thought all 3 were the same, but you'll notice that the brown one fit quite well. That's the one I used for the correlation, but the others did not. So if I'm going to do this correctly, then I have to do all 3 images individually and bring them in separately. There's not a one-to-one correlation between those 3 images. But you can see then the EBSD is now capable, or OIM is capable of showing this color map on top of whatever kind of map you want to make, whether that be image quality, confidence index, some other type of map. So you can do that. So in summary, I hope I've given you a lot of examples, but we can do various analyses of the data too. So you can do scalar textures, pair charts, and highlighting. So with what we call a scalar texture is we can take any scalar value and we can calculate the average for each bin in orientation space. In this case, we're just using Euler angles. And then from that, you basically calculate, it's almost like an ODF, an orientation distribution function. But instead, at each bin, we actually have the average value for the CMB. And from that, then you can make an inverse pole figure like I've shown here, or a pole figure. So you can see there's a little bit, this is the CL data. There is some correlation there. You see a bit of a peak in the IPF, but I haven't searched around to see if there's more detailed correlations. But you know, that's not a huge difference. The max there is 8 for the CL data, whereas the minimum value is somewhere around 7.5. So it's not a huge difference. Here's the micro-hardness. This is kind of interesting. I wanted to see if there's a correlation. It appeared to be between the kernel average misorientation and the micro-hardness data. So you can see the kernel average misorientation on the horizontal axis of our pair chart. And then the hardness data, there's the vertical axis. And you do see a correlation when the cams are low. the hardness is smaller, and then as the cams increase, the hardness increases, and that is expected behavior, so that's always a relief. But just to show that the correlation can be used then with these pair charts in OIM. Now, obviously, you can do any kind of highlighting, so you can highlight from the CMV chart. In this case, I've got the hardness on the horizontal axis, and then just the distribution number fraction on the vertical axis. So in this case, I clicked and selected the whole plot, so it creates a gradient from blue to red, and then I have above, I have the pole figure, the discrete normal direction pole figure for my data, and it's trying to see if there's any correlation between the micro-hardness data and that. And I'm not sure there is, but it shows you that highlighting can help you find any correlations that might be existing in the data pretty quickly. Oh my goodness. So in conclusion, I hope I've shown you the range of tools we have, both for correlating EBSD with other imaging modalities, as well as analyzing that afterwards. And as I've pointed out before, the spatial resolution of the correlated data is always matched to that EBSD data grid. So the EBSD data grid holds it. We don't create some kind of super grid, but it's just attached to the EBSD data grid. But it does, but the nice thing is we can link those CMVs into the whole suite of data analysis tools. So you could do texture analysis, the highlighting, the pair correlation charts that we've had for quite a while, but this really brings them in. We haven't used them a lot, but I think with this, it really makes it so they're really useful. So I hope that shows you exact what we can do, and that you'll be excited to get 9.1 once it's released, hopefully here within a month or two. So we'll see how it goes. But I especially want to thank the following for all the data and images they have given me to do this presentation. Guillaume Willet in France, Chavan at the University of Hyderabad, Daniel Aburas at BAM in Germany, Tim Ruggles, who's at Sandia National Lab, Marco Witte at the Salzgitter Mannesmann Forschung, Z Chen and Sam Daly at University of California, Santa Barbara, and David Fuller at Brigham Young University, as well as my colleagues Matt Nowell and René Declue, who have been really helpful in getting me some of the nice images that I was able to show you today. So thanks to all those folks, and hopefully you'll be able to create the same images yourself and see yourself on one of my slides in the future, or more importantly, in a nice paper published in the literature. So thanks for that. If you have any questions, please submit them. And I guess I can take a look at the questions here, see if any have come through so far. And I didn't see any, so thanks for listening in, and I hope that's helpful. If you do have questions, don't be afraid to ask, and I'll turn the time back over to Jonathan.
Thank you everyone for attending today's webinar. There'll be an on-demand version available tomorrow. It'll be via our YouTube channel. You'll also get an email for attending today. Thank you so much for your attention. Thank you, Stu.
Thank you.